Tacrolimus. Tacrolimus, also FK506 or fujimycin, is an immunosuppressive drug whose main use is after organ transplant to reduce the activity of the patient's immune system and so the risk of organ rejection. It is also used in topical preparation in treatment of severe atopic dermatitis, severe refractory uveitis after bone marrow transplants, and the skin condition vitiligo. It was discovered in 1984 from the fermentation broth of a Japanese soil sample that contained the bacteria Streptomyces tuxibannus. Tacrolimus is chemically known as a microlide. It reduces peptidyl propyl isomerase activity by binding to the immunophilin FKBP12, creating a new complex. This FKBP12 FK506 complex inhibits calcineurin, which inhibits T lymphocyte signal transduction and IL-2 transcription. A promising approach to determine the biological effects of immunosuppressive drugs may be the measurement of intracellular signaling pathway activation. Phosphospecific flow cytometry of the intracellular signaling molecules P38, MAPK, ERK, and AKT is a promising technique to monitor the pharmacodynamic effects of immunosuppressive drugs in whole blood of kidney transplant patients. Tacrolimus inhibits calcineurin and therefore T cell proliferation by arresting the cell cycle GO and G1 in a similar manner to cyclosporin. After binding to a receptor protein called FK binding 12, the complex binds to calcineurin and inhibits CA2 positive dependent calcineurin activation. Tacrolimus also inhibits the MAPK pathway in a similar manner to cyclosporin. Unlike cyclosporin, tacrolimus does not stimulate production of TGF-beta. Calcineurin inhibitor tacrolimus, its potent inhibitory effects on adaptive immunity, is one of the predominant anti-rejection drugs used nowadays. A proper innate immune response is of great importance in clearing bacterial infections and mainly dependent on pathogen recognition by pattern recognition receptors, such as toll-like receptors. The mechanism of action in atopic dermatitis is not known. While the following have been observed, the clinical significance of these observations in atopic dermatitis is not known. It has been demonstrated with tacrolimus inhibits T lymphocyte activation by first binding to an intracellular protein. A complex of tacrolimus, calcium, calmodulin, and calcineurin is then formed, and the phosphatase activity of calcineurin is inhibited. This prevents the dephosphorylation and translocation of nuclear factor-activated T cells, a nuclear component thought to initiate gene transcription for the formation of lymphokines. Tacrolimus also inhibits the transcription for genes which encode IL-3, 4, 5, GM-CSF, and TNF, and all are which involved in early stages of T cell activation. Additionally, tacrolimus has been known to inhibit the release of preformed mediators from skin mast cells and basophils to downregulate the expression of FCERI on Langerhans cells. Calcineurin is a negative regulator of TLR-mediated activation pathway. By inhibiting the adapter proteins MyD88 and TRIF18, therefore, we treated macrophages either with solvent or tacrolimus and determined activation of TLR-mediated pathway. Both stimulations with LPS or tacrolimus led to phosphorylation of IKB alpha and nuclear translocation of the NFB-KB into the nucleus as assessed by Western blot immunofluorescence staining, respectively. In line with these results, TNF-alpha mRNA was upregulated by LPS and tacrolimus stimulation in macrophages and granulocytes. Thus, tacrolimus alone activate NF-kB pathway and hence may influence TLR pathway in myeloid cells. Maintenance therapies vary by type of organ, institutional preference, and organ recipient demographics. A multimodal approach is commonly employed to prevent rejection by blocking immune responses through several pathways. Modifying immunosuppression regimens to selectively inhibit effector and memory T cells while permitting TREG development, survival, and function could theoretically allow for minimization of immunosuppressive drugs and favor tolerance induction. Current knowledge of TREG biology reveals numerous distinctions between TREG and T-cons that may be targeted therapeutically to favor TREG. Many of these distinctions, such as IL-2 responsiveness, CD28 and mTOR dependence, CNI sensitivity, and resistance to lymphodepletion, are not absolute but quantitative. Thus, selection of TREG-friendly immunosuppressive regimens not only has to consider which drugs to use, but also what dose to apply. Many immunosuppressive drugs currently used in transplant patients are compatible with TREG at lower doses. It is possible that a combination of multiple immunosuppressant drugs at low dose would be better to support TREG while adequately preventing rejection while minimizing toxicity. When evaluating new immunosuppressive regimens, close monitoring of the numbers, activation status, and the function of T-cons versus TREG, in addition to clinical outcomes, will help to enrich our knowledge and guide future development of tolerogenic therapies for transplantation. It is important to note that high proportion of TREG needed to induce transplant tolerance in preclinical models is not likely achievable by titrating doses of immunosuppressive drugs alone. Combining TREG cell therapy, attenuation of effector responses, and TREG-supported immunosuppression may be needed to induce tolerance.